Hello. It's nice to have you all with us again today after a couple of months, especially our anniversary month, April, filled with various events organized by our volunteers. And I hope you had been enjoying everything that we have been releasing, from blogs to live discussions, panel events, so on and so forth, together with the competitions that we had on Twitter. So as you all probably know by now, our theme this month is focusing on cancer. And this evening, we are privileged to have a professor from Manchester with us. And this will be our first live session of the month. So as I mentioned, Professor Stephen Taylor is with us for our Q&A session this evening. And we will be focusing on his research strategies for developing new therapies for ovarian cancer. So before starting to learn about this interesting topic, I would like to introduce our structure to those who are not, who might be new to our platform. So in the next half an hour or so, Steve will be answering our questions. And after this, we will have a 10-minute comfort break and resume back for a Q&A session. So during the discussion, during the break, if you have any questions, please feel free to post them through our social media accounts, through email, or on our YouTube channel using the chat function. So during the break, I will collate all of these questions and I will be asking these on your behalf. So like I say, feel free to post any follow-up questions if required as well. So I would like to briefly introduce Steve. So Steve is a professor of cell biology at the Manchester Cancer Research Centre. So after completing his degree at the University of Manchester, he has moved to Oxford University for his PhD and continued his postdoctoral studies at Harvard Medical School in the States. So after his return to Manchester, he has established his independent group, and we're lucky to have him since then. So Steve, it's an absolute pleasure to have you with us today. Thank you for agreeing to be here, and we look forward to hearing about you and your research.
Great. Well, thanks for the invitation. I'm a bit nervous. I feel like I'm on Mastermind.
Oh, no. Black chair. No, we haven't got any hard questions. It's all to understand about your research and, how you ended up where you are today and similar things. So I would like to first start by asking, so have you always worked in the field of cancer research? I know you've been around, but have you always been in cancer research?
No, not really. Going back to my early interests, it was really about basic biology of sort of genomes and chromosomes and then cell division, and really just understanding how it all works at a basic level. And I guess it then became obvious that what we're working on would have implications and was relevant to cancer. But I guess we were, my interests were a bit sort of, again, still that sort of mechanistic end of the spectrum. And I was rather agnostic in terms of a type of cancer. And I'd say it wasn't until 2015 that we really became very focused on ovarian cancer. So I guess it evolved from basic mechanisms to thinking about cancer in general terms, to then applying it more specifically to ovarian cancer. And that's been a 20-year sort of process.
Okay. And obviously I'm a cancer researcher as well, and I find it fascinating. But what makes cancer research such an interesting field for you?
So again, I guess that the real fundamental interest still stems from the biology, the underlying biology. So we know that when cells divide, they have to duplicate and segregate their genomes such that the two daughter cells each inherit a complete copy of the genetic material required for further growth and development. And that's just a fundamentally fascinating process, because of course it's happening in our bodies all the time. But from a cancer perspective, what we know is that sometimes it does go wrong. And that's what gives rise to these very abnormal genomes that cancer cells have. And so, yeah, it's really, I guess the interest stems from what do we do with that knowledge that we've gained from the basic biology? How do we then extrapolate that or translate that into something useful in terms of understanding tumor biology and also developing novel therapies if possible? So that's the interest is a continuum from that sort of basic through into the cancer biology.
Okay, that's great. So you told us that you have been focusing on ovarian cancer in the recent years of your career. So can you tell us a bit about ovarian cancer, how common it is, what are the risk factors for it?
Sure. So yeah, in the UK, there's probably about 7,000 or so new cases per year. Long-term survival rates are not great. They're not terrible. They're not great. So, there's a lot of work to do if we're to improve outcomes for women with this particular disease. In terms of risk factors and so on, I mean, again, it starts to get a bit more complicated because there's different types of ovarian cancer. And the one that we're most interested in is high-grade serous ovarian cancer, which is the most prevalent and the most deadly form of the disease. And the challenge there is that it tends to develop quietly. Sometimes, the newspapers, they call it the silent killer. And so when patients present with the disease, it's already quite well advanced. It's already undergone local metastatic spread. And so treatment options then become quite challenging. So the surgeons will typically try to remove as much of the tumor as possible. But then it's the chemotherapy. The chemotherapy is then what will then keep that residual disease at bay for as long as possible. But then unfortunately, patients will relapse. That would then be followed by more chemotherapy. And again, by using, the clinicians can do a fantastic job using different combinations of chemotherapy. There are some really new exciting agents coming along, which I can talk about. But by trying all those different combinations, again, that can keep the disease under control for reasonable periods of time. But in a lot of cases, drug-resistant disease will eventually emerge. And so eventually those women will die because of the disease. Okay. And that's really what I guess we're kind of interested in, is to really improve outcomes. It is going to come down to better and better systemic therapy. So better chemotherapies, smarter chemotherapies. So there's the traditional chemotherapies, but now there's the next generation of more targeted therapies. PARP inhibitors is a class of drugs that are very exciting for a subset of women with ovarian cancer. And those are having real profound effects in the clinic. And it's really about, what other options have we got for the other women who won't benefit from PARP inhibitors?
Okay, so you mentioned that, about traditional and next generation chemotherapy agents. Apart from the molecules that they're targeting, what is different about them? Why are they far better than the conventional therapies?
Yeah, that's a good question. So I guess, the traditional chemotherapy, which have been developed and optimized over many, many years, are in effect poisons. And they're very good at killing cancer cells, but they're also very good at killing normal cells as well. And of course, that's why you get such toxicity with them. And so the tumor selectivity, if you like, is not great. Yes, it's in the tumor cells, but they're also hitting normal cells. And this is why the oncologist has to be very careful with the dosing, the scheduling, the timing, managing the side effects to try and maximize that anti-tumor benefit. And of course, the dream scenario is to come up with new strategies that are very toxic against the tumor cells, but really don't touch the normal cells at all. And that's the sort of the dream scenario. And in order to develop therapeutic strategies along those lines, you need to understand about the biology of the specific tumors to identify vulnerabilities, ways that you can then target them. And so what we now know about ovarian cancers is that about maybe up to as many as 50% of them have homologous recombination defects. So they have a type of DNA damage repair defect. And this makes them particularly vulnerable to drugs that inhibit an enzyme called PARP1 and PARP2. Now, those drugs have virtually no effect on normal cells because normal cells have other ways, redundant pathways that they can cope with being exposed to these drugs. But tumor cells are particularly sensitive to these PARP inhibitors, resulting in what we call a synthetic lethality relationship. And that's really very exciting. And these drugs are very well tolerated. The side effect profiles are relatively very, very benign, and they, in the right patients, have very profound anti-tumor effects. So this is really, really very exciting.
Okay, thank you. That was very detailed. So in terms of then the how ovarian cancer develops, do you see common pathways in different types of ovarian cancers, or do you see very different pathways involved in subtypes of ovarian cancer?
Yes, so that's right. So there are about there are 5 main types of ovarian cancer, and yes, it's exactly as you say. There are different pathways. So different genes are affected, different genes are mutated, but then even within one subtype. So let's go back to high-grade serous, which is the one we're most interested in. Even within that one subtype, there are some common features. So the very famous tumor suppressor gene called P53, that's mutated in virtually all of them. But then beyond that, there's not much common between a lot of them, other than they all have very abnormal genomes. They all show high levels of what we call chromosome instability. They're constantly missegregating chromosomes, they're rearranging their chromosomes. But even within the concept of chromosome instability, there seem to be different flavors of chromosome instability. And at the moment, we're really only getting to grips with the understanding of that. There's still an awful lot to learn. So we know something about the very early events, and we know that later on there's a whole spectrum of phenotypes and behaviors, but quite how we connect the dots through different pathways to get to those different final tumors is still a big mystery.
Okay. So do you think, is ovarian cancer, would you consider it more of a genetic sort of type of cancer, or is it related to environmental factors? And if it's related to environmental factors, are there any known factors which are putting people at increased risk?
Yeah, I probably wouldn't want to talk too much about that because that's not quite my area of expertise. I mean, I think there have been over the years, certain things linked to it. Certainly the genetic background. Obviously, if you one of the big fundamental risk factors is inheriting a defective BRCA gene, for example, which is a gene which is involved in homologous recombination. So that is that's an enormous risk factor. And I think it's widely accepted now that sort of inflammatory environments and so on are probably playing important parts in the early stages of tumorigenesis. But what exactly those are, I think isn't really clear.
Okay. Okay, fair enough. Thank you. So I'm actually interested to find a bit more about your living biobank strategy for developing better treatments, better therapies for women. Can you tell us a bit about what a living biobank is, how this idea came about, and how actually you're keeping the tissues alive while you're doing experiments?
Yeah. So our living biobank has been a major undertaking in the last 5 years, and I think it's something that we're incredibly proud of. I think so. Going back to your very early questions about my interests and moving from sort of basic mechanisms and understanding how cells work, and then wanting to apply that in a more clinically relevant context, I think we chose to work on ovarian cancer, and what I really wanted to do was test out some of the ideas and the hypotheses and the things we've been discovering in the lab, because I wanted to try those in a more clinical, in material closer to the patient. And so the question was, could we get patient biopsies, and could we bring those biopsies into the lab and study them and test some of those hypotheses? Now, of course, people do this all the time. You take a tumor, the surgeon will take a tumor and they'll plonk it into some, fixative, and then, they slice it up, and that's how you do all of the histopathology and so on. And the pathologists make their diagnoses and so on. But we wanted to do something a bit different. Like you say, we wanted to keep the tissue alive. We want to bring it into the lab and ask, could we do experiments on the tissue and on the cells in the lab? So when I first decided that I wanted to do this, I thought, how hard can it be, right? I mean, tumor cells, they grow like weeds. We've been working on established cancer cell lines for years and years and years. We've had cell lines established in the 60s and 70s. They grow very easily in the lab. The limitation is surely just access to samples. Once you've got access to samples, it's going to be easy. I was rather naive. Turns out it was a lot harder than we imagined. But to cut a long story short, we ended up figuring out a way that we could bring the biopsies from the Christie Hospital to the lab. We could then isolate the cells, plate those in tissue culture, and through a process of looking after the cells, we could separate out the tumor cells from the non-tumor cells. And so we ended up with paired samples, tumor cells and non-tumor cells from the same patients. And the fantastic thing is that they proliferate beautifully in cell culture. We've now purified them away from all of the complexity of the original tumor. So we're just looking at the tumor cells. And so now we can really start to hone in on tumor intrinsic properties intrinsic to the tumor cells, without having to worry about the microenvironment and anything like that. And so we've now collected, probably over hundreds and hundreds of samples from the Christie Hospital. One of the advantages, if you like, of working on ovarian cancer is that for a lot of tumor types, if you want to collect samples, you need to be friends with a surgeon and the surgeon to provide you with the samples. But with ovarian cancer, what happens is very often when we get a buildup of fluid in the peritoneal cavity, and that fluid is uncomfortable, and so they'll come into the clinic to have it drained and removed. And normally it would just go down the sink, be thrown away, but it's full of tumor cells. So if you can keep the fluid and isolate the cells from it, it's actually a very rich source of tumor cells.
Okay.
And so what that means is it's actually quite not easy. There's still a big logistical process to play, but it's relatively easy to get samples from ovarian cancer patients. But the other important thing is it means we can get multiple samples throughout their treatment. So what we're now starting to build is samples where we've got a biopsy from the same patient before they have chemotherapy, while they're on chemotherapy, and after chemotherapy when they then relapse. So we're getting the so-called longitudinal samples that are giving us a huge amount of information about individual patients and how their disease evolves during that process.
Okay, that's very interesting. So you mentioned about using the cells in your experiments. Can you tell us what type of experiments you're actually doing on them? Are you treating them with new therapeutics? Are you doing experiments to understand their biology?
Exactly, both. So the great thing is we can do chemotherapy in the lab. So again, going back to the traditional biopsy pipeline, is you end up with, you fix the tissue, it's dead. You can extract the DNA and you can look at all the mutations, you can extract the proteins, you can analyze biochemically, but you can't do experiments to ask how the cells would behave. So the ability to have proliferating cell cultures just opens up a whole new world of analysis that one can do on these patient biopsies. So, yeah, so we've been, going back to the basic biology, we've been analyzing them by time-lapse microscopy, watching them divide, seeing how they divide. And, as you might expect for this particular disease, it's incredibly chromosomally unstable. The divisions that they go through are highly abnormal. It really makes you wonder how they stay alive sometimes. So we've been studying cell division very carefully in these cells. And then, yeah, we as I mentioned, we can do chemotherapy in the lab. So we can take the chemotherapy drugs that the women were being treated with during their course, and we can recapitulate that in the lab. And we can see which drugs they're resistant to, which drugs they're sensitive to. And then we've also got a pipeline of new drugs, drugs which have not yet gone into patients. New ideas, new targets, new molecules, new small molecule inhibitors becoming available. And so we can test those in our biobank, ask the question, which cells are sensitive and which cells are resistant? And then this will give us a clue as to whether these new therapies might be effective in this disease or not. Okay. So yeah, so again, long answer, but basically, yeah, we can do really nice cell biology to understand mechanistic issues, and we can do
and we lost you there for a couple of seconds. Would you be able to repeat your last sentence, please?
Sure. Yeah, just I was just summarizing to say that we can, with our proliferative cultures,
yeah,
we can do both mechanistic cell biology, and we can do sort of in vitro chemotherapy. So it's very exciting.
Okay, great. Really interesting and really exciting. So obviously you have already mentioned that there are difficulties actually obtaining the cells, keeping them alive, and making sure that they sort of survive. So what were the main challenges, though? Yes, these are the problems that you have, but how were these affecting your research, and how did you actually overcome these difficulties?
Yes, so good question. I think the sort of the dirty secret of cancer biology that we discovered, which we didn't really know about, was that when you isolate a biopsy from a patient, and this, whether it's a solid biopsy or an ascites, it'll be a mixture of tumor cells and non-tumor cells, as I mentioned earlier. And what we're taught is that tumor cells are immortal. They've overcome this mortality, and they'll proliferate and they'll keep on growing. Whereas the non-tumor cells are mortal, and eventually they'll go through a certain number of cell divisions and they die. And this is certainly true, but when you take a biopsy and put it into cell culture like we normally do, the paradox is the non-tumor cells grow like crazy, and the tumor cells don't grow at all. This just makes no sense. But this is a problem that pervades the cancer biology field. And I think what it tells us is that the standard cell culture conditions we use for growing established cell lines, they work on established cell lines that were generated decades ago, but they're not appropriate for the various different tumor types we're trying to study now and trying to generate fresh cultures for. So really, what we didn't have was the right media that would allow, the right culture media that would allow the tumor cells to grow, but perhaps not the stromal cells. So we set about trying to identify the cell culture media, the cell culture conditions that would allow the tumor cells to grow. But all the work was done for us because a professor he's now in New York, he was in Miami at the time, Professor Tan Inche, he published a paper in 2015, which is a fascinating paper. It took him 10 years to figure out how to grow primary ovarian cancer cells in the lab. And this goes back to what I was saying earlier about how I was naive. I thought it would be easy. Once we could get the samples, we would just bring them into the lab and they would grow. naivety was that, yeah, it took him 10 years to figure out how to do it. But fortunately, once he'd figured it out, we could simply recapitulate his recipe. And the first time we tried it, it worked. So it was a major breakthrough, just being able to reproduce his recipe and get his cells to grow, get our cells to grow in his recipe. And once we did that, we never looked back. From that point, our biobank has been growing and growing. So that was the major technical hurdle that we had to overcome, was identifying the right tissue culture media to allow the cells to grow. But there's one other very important ingredient, and of course it would be a mistake for me not to acknowledge this. Of course, it's the people that do it, and in particular in my lab, it's one of my postdoctoral research associates, Louisa Nelson. She's the one who set all of this up from scratch, and she's the one who, day in, day out, collects the samples and sets them up in the lab and sets up these separate cultures. And she's developed the skills to enable her to do that. So I think it's really the combination of the recipe and the individual that has allowed us to make this a really big success. We're now in a position where we can teach other people to do it, but I think it was that combination was very important to getting this whole thing off the ground.
Fantastic. Very good to hear, though, and very interesting as well. So we have done, if you have followed our program, you will know that in April we have actually focused on involvement of public in research. And I would like to ask you, do you find that patients are normally keen to take part in your research?
Yeah, so obviously all of our patients are consented, and what we find is that the uptake or the willingness to contribute samples is very high. And so that's absolutely fantastic. And I think, again, that's not down to us. I think that's down to the doctors, the research nurses, the individuals that the patients interact with on a day-to-day basis. They're fantastic at developing those relationships, which then allows the patients to be confident and willing to contribute their samples. So yeah, no, we're very, very lucky that the patients are willing to be involved in research. But again, like I say, it's the healthcare professionals who interact with them on a day-to-day basis that are really the important ones for developing those relationships to allow that to happen. We've also been a little bit involved with patient advocate groups, and that's also been very helpful because just getting the perspective. As I mentioned earlier, these PARP inhibitors are a very exciting new area in ovarian cancer, but they're only going to be useful in up to 50% of the patients, let's say. And in talking to the patient advocate groups, there's a feeling that from the others that they're somehow a bit left behind. And they're thinking, well, there's all this excitement for those individuals, but what are the new developments for us? And so I think, interacting with patient advocate groups does help you think about your vision and focus and where the clinical challenges lie. I think that's also very important.
Yeah, certainly. Yeah, definitely. Right, so I have made you to talk for 30 minutes non-stop, so I think it's time to give you a break. And during the break, like I said, we will collate the questions. So let's meet in 10 minutes at 7:10. Thank you, everyone. Okay, great. So thanks very much, everyone, for waiting for us and for submitting your questions. So I've got my phone next to me, so I'm going to read the questions from the YouTube channel, Steve, if that's all right with you. So we have a question from our audience. They're asking, is the progress made with the culture of ovarian cancer biopsies transferable to other forms of cancer?
Just got some interference on my computer. Yeah, good question. So as I mentioned earlier, the magic recipe that we're using was developed by this professor in Miami. And he developed that from another media which he'd used for breast cancer cells. But I think that works reasonably well, but certainly his ovarian one works a lot better. But yeah, you can't take this media and use it for other cancer subtypes. It's very specialized to ovarian cancer. But other scientists, other researchers are developing similar methodologies for other cancer subtypes. So colon cancers, for example, you can grow them in sort of 3D organoid type structures. Those work quite well. Breast cancers also can be grown in these 3D organoids. So in the last, 5-10 years or so, this is a rapidly emerging field. There are other cancer subtypes where it's just, people haven't made the breakthrough yet, and it's just really difficult and really challenging to get primary cultures to work at all. So we're certainly at the forefront in that sense.
Okay, great. So does it mean that these sort of biobanks would enable patients to receive personalized therapy in future?
Yeah, I mean that would be fantastic, and that is the sort of the fantasy vision, if you like. If we could bring the sample into the lab and very quickly generate the culture and test it with a variety of drugs, and we could do that to a high enough standard that indeed we'd be able to then go back to the doctors and say, look, these drugs work, these ones don't. That would be a way of using the biobank samples as a patient avatar, as a readout, as a mimic for the actual patient tumor. Yeah, I think that would be, like I said, that's the fantasy in the long term. We're not there just yet, but yeah, maybe one day that'd be pretty spectacular.
Okay, great. Thanks very much for that. So one of the other things that I was wondering though, can you tell our audience a bit more about, when you develop new therapies for targeting ovarian cancer, what is it that you're actually targeting?
So like with the PARP inhibitors, for example.
Yeah.
So the PARP inhibitors themselves target an enzyme, or a few enzymes called PARP1 and PARP2, and they're required for DNA damage repair pathways. I guess the easiest way to explain it is that cells have a variety of DNA damage repair pathways, and these are sort of some overlapping. There's redundancy, really, so that if anything goes a little bit wrong with any one pathway, the cell can still cope with that. But what we find with a lot of tumor cells is that they have defects in these DNA repair pathways, and a subset of ovarian cancers in particular have a defect in a process called homologous recombination, and this is very often brought about by mutations in genes I mentioned earlier, BRCA1 and BRCA2. So if you have mutations in this pathway, you now become absolutely dependent on this other pathway. So if you're mutated here and now you inhibit this pathway, now you collapse the system and the cell cannot repair any damaged DNA and it will eventually die. So what you're targeting is not so much the mutated proteins that are driving the cancer. You're targeting a subset of proteins that the cell is now absolutely dependent on because it's got these mutations over here. Whereas in normal cells, if you inhibit these pathways, the cells can survive just fine because they've got the redundant pathways over there. So this goes back to the synthetic lethality relationship I was talking about earlier. Does that help?
Yeah, definitely. Thanks very much. And in terms of developing therapies and delivering them to the areas or making them specific for the tumor cells, I know that you had some research on sperm bots to deliver chemotherapy to the specific areas. Could you tell us a bit more about this?
Yeah, so that's not the sperm bots isn't directly my research. That's research by a junior faculty in the department, Christine Schmidt. And we provided her with some samples to test out her ideas. It's kind of a that really is next generation ideas. I guess one of the fundamental principles is it goes back to the toxicity issue, that there's always going to be toxicity side effects, because with these systemic therapies, you're infusing or you're taking a tablet or something, and so all the cells in your body are being exposed to the drug. A much smarter way of doing things would be if you could physically target the drug to the cancer cells such that it doesn't actually even interact with any of the normal cells. And so there's a lot of work, around the world going in on drug delivery mechanisms. Can you target a drug physically such that it's only transported to the tumor cells and therefore can't harm normal cells? And there's all sorts of fancy ways of doing that. Viral sort of vectors to try and deliver suicide genes to cancer cells is another way of doing it. There's all sorts of amazing stuff like that going on. The sperm bots is kind of like a real super advanced idea to try and think about that. Can we use nanomachines to deliver chemotherapy agents in a highly specialized way? Now, whether that will ever become a therapy is, who knows? That's 20 years down the line. But as an intellectual idea, it's really quite clever. And I think it's one of those ideas that makes you think, actually, maybe some crazy things are possible. And if you just imagine bigger, we might be able to do some really special things.
Yeah, great. Lovely. I think this is the end of the questions. So I would like to again thank you for your efforts today, for your time, for being with us. And, it was really interesting, and I'm sure the audience has enjoyed it as well. Thank you.
Well, yeah, thanks very much for the invitation. I hope people enjoyed it. Some great questions. Thanks again. Thanks very much.
Lovely. Thank you. So see you everyone next week on Tuesday with our live talk with Dr. Isabel Pires. And thanks very much for your interest.